Hi, everyone, and thanks for coming. I'm Carolyn Klinge. I'm a professor of biochemistry and molecular genetics. My lab and office are right upstairs here in the Delia Baxter building, and I'm very glad that Linda Petrie, who is now a postdoc working on this project and previously was a PhD student in my lab, and she can probably answer some of the questions about the M6A analysis that we'll come to about halfway through this presentation today. So I'm delighted to share with you some of the research that we're doing on the role of M6A in RNA transcripts in endocrine resistance in breast cancer cells. And I have no disclosures today. So on the seminar sheet, I had to put some learning objectives. So these are the 4 learning objectives for this talk, but I just thought, why don't we see what we're really talking about in brief here and what you'll hear over the course of the next 45 minutes or so. And so N6 methyladenosine is an epitranscriptomic mark, a chemical modification of transcribed RNA, and it's the most abundant modification in mRNA. And it's applied to RNA by the metal-3 transferase complex. So that's the writer complex. Metal-3 is the methyltransferase that takes the methyl group from S-adenosylmethionine and places it on the the N6 of adenosine. And this occurs on transcribing RNA, so it actually happens co-transcriptionally as the RNA is exiting from the RNA polymerase, but it also occurs on primary microRNA transcripts. And the M6A mark is recognized by a number of different reader proteins that we'll look at in a few slides. But one of the reader proteins is hnRNA2B1. And that interacts with the Drosha complex to increase processing of certain primary microRNAs into the precursor microRNAs that are then transported into the cytoplasm where they interact as mature microRNAs with the hnRNA2B1 with the RISC complex after being processed to the mature microRNAs by the dicer complex. And what you'll see in the course of this presentation is we know there's M6A marks on certain microRNAs that seem to be regulated by hnRNA2B1 that actually target genes that are critical in the serine synthesis pathway, and these are PSAT1 and PHGDH. We also know that we can find M6A marks on mRNA transcripts by direct RNA sequencing through latest technology from Oxford Nanopore, nanopore RNA sequencing. And one of the transcripts that we identified as having differential expression as well as differential number of M6As is amigo-2. So we'll talk about amigo-2 and its role in apoptosis sensitivity in endocrine resistance and endocrine sensitivity. So as I've said, what epitranscriptomics is chemical modifications of transcribed RNA. And this is all transcribed RNA. So the most abundant RNAs in the cell are tRNAs and ribosomal RNAs, and they are the most heavily modified. And you can see a number of modifications that occur on all 4 nucleosides in RNA in this slide. But what about mRNA? Well, there's 13 known modifications of mRNA, 11 of which are depicted here, including N6 methyladenosine. But it's not that every A is modified because the A has to be in a certain sequence that we're going to look at. It's called a Drash motif. So only 0.1 to 0.4% of A's are exposed to this M6A modification, and with an average of 3 to 5 M6A sites per RNA transcript. And the thinking really is that M6A destabilizes RNA, causes conformational changes in the RNA and affects stability. But it really depends on those reader proteins that recognize that M6A mark. I got interested in M6A and epitranscriptomics after reading a couple of papers in 2015, but I'm not the only person who thinks that M6A and epitranscriptomics is really interesting, as you can see by the number of publications with M6A in PubMed per year over time. And here's M6A in breast cancer. And it seems to be, the interest seems to be leveling off. Well, as promised, we're going to look at the writers, readers and erasers of the M6A mark. And this is a lovely image that Belinda created as part of her dissertation defense, and this was in a publication, a review that we wrote for JME last year. So here's the adenosine being methylated by the metal 3 complex, and it's associated with a number of proteins that are involved in the process, including metal 14, which is a heterodimer partner for metal 3. And even though it says it's metal, it's not really a methyltransferase. It's metal 3 that's the methyltransferase. And then the M6A mark is read by a number of readers, both within the nucleus as well as out in the cytoplasm. And depending on the interaction and reader, then M6A marks are involved in alternative splicing events in, as I said, increasing the processing of primary to precursor microRNAs, involved in export from the nucleus. And again, depending on the reader, we can have an increase in mRNA decay or stabilization of that transcript and more translation into protein or protein silencing. Now, the M6A mark is dynamic. So not only does the metal 3 transferase complex add the methyl group to form M6A, but we also have 2 erasers. Now, the reason the FTO is, which means these are demethylases that take that methyl group off of the A, so it goes back to A. And this A is always in the Dratch motif. And the Dratch motif, you can see what the nucleosides are here, but it's invariably the A is in the center and it's followed by a C. And that's what's recognized by metal 3. So the ALK-BH5 and FTO can remove that methyl group, and then it just goes back to an A, in which case these readers won't be interacting with that transcript at that site in the absence of that methyl group. So what do we care about this? What's the clinical relevance? Well, we all know that endocrine therapy is a continuing problem for breast cancer patients with estrogen receptor positive tumors, even with the advent of a number of CDK4/6 inhibitors, PARP inhibitors, and other modulators of downstream effects and pathways of endocrine resistance. So 33 to 40% of patients who are on endocrine therapies will develop acquired endocrine resistance. So my question was, well, what's the role of transcriptomics in acquired endocrine resistance in breast cancer? And to address that question, we use a cell line model system, starting with MCF7, which are luminal A estrogen receptor positive breast cancer. So they require estradiol for their growth, and their growth inhibited by the anti-estrogen or endocrine therapy tamoxifen, as well as fulvestrant, a selective estrogen receptor degrader. T47D is also an estradiol-dependent tamoxifen-sensitive cell line. And then modeling breast cancer progression of patients who are on tamoxifen or aromatase inhibitors, we have tamoxifen-resistant cells. These are LCC9 and LY2. And I got these cell lines from Robert Clark when he was still at Georgetown University. He's now at the University of Minnesota. So I first looked at the expression of all the readers, writers, and erasers in a data set that we had of RNA-seq between MCF7 endocrine sensitive versus LCC9 endocrine therapy resistant breast cancer cells. And what I found was that there was an increase in hnRNA2b1 in the LCC9 endocrine therapy resistant cell line compared to MCF7. And hnRNA2b1 was higher in breast tumors compared to normal breast samples, and higher, now we're going to call it A2b1 because it's a lot to say hnRNA2b1 all the time, so we'll just shorten it to A2b1. High A2b1 was associated with a lower relapse-free survival in ER-positive breast cancer patients. So if A2b1 is involved in endocrine resistance, can we decrease A2b1 and restore endocrine sensitivity in LCC9 and LY2 cells? So in these experiments, we transfected either LCC9 endocrine therapy resistance or LY2 endocrine therapy resistant cells with either an SI control or SI2A2B1. We can see the loss of A2B1 protein expression in these cells. And then we performed MTT assays for 5 days, treating the cells either with a vehicle control, DMSO, or with 2 different concentrations of 4-hydroxytamoxifen or 100 nanomolar fulvestrant, the selective estrogen receptor degrader. And what you can see is that with the combination of SI-A2B1, we see sensitivity in terms of inhibition of cell viability in the presence of treatment with 4-hydroxytamoxifen or fulvestrant, both in LCC9 and LY2 cells. And so we were able to publish a paper using these data showing a role for A2B1 in endocrine therapy resistance. The next thing we wanted to do, because A2B1 was known to be involved in that primary microRNA to precursor RNA processing in the nucleus in association with M6A deposition. What if we overexpress A2B1 in endocrine therapy sensitive MCF cells, which will convert them phenotypically to endocrine therapy resistant, so they're no longer growth inhibited by tamoxifen or fulvestrant? What are the microRNAs that are regulated? So we overexpressed at four times expression hnRNA2B1 in MCF7 cells and did RNA-seq. And Dr. Eric Rushka, who was a professor in biochemistry and molecular genetics, analyzed these data, and we identified differential expression of microRNAs at different time points after overexpression of hnRNA2B1. And one of the pathways that we found associated with these differentially regulated microRNAs with A2B1 overexpression was the serine synthesis pathway. And that struck a chord with me because Dr. Brian Klinge, who's also in biochemistry and molecular genetics, has been studying the serine synthesis pathway. And his PhD student, Stephanie Metcalf, had looked at PHGDH and PSAT1 protein expression in the LCC9 and LY2 endocrine therapy resistant cells and saw that those 2 enzymes, which are key enzymes in the serine synthesis pathway, were upregulated. I neglected to say that the serine synthesis pathway comes off of glycolysis, every biochemist's favorite pathway, and PHGDH and PSAT1 are the rate-limiting enzymes in the serine synthesis pathway. And we also found that high PSAT1 and high PHGDH are associated with lower metastasis-free survival in breast cancer patients. So we looked at our A2B1 stably overexpressing MCF7 cells at PSAT1 and PHGDH expression, and similar to what Stephanie Metcalf and Brian Klinge had already shown for LCC9 and LY2 cells, we see a higher expression of PSAT1 and PHGDH protein in these endocrine therapy resistant cell lines. So we hypothesized that specific ATB1 downregulated microRNAs target and suppress PSAT1 and PHGDH expression. So Belinda Petrie looked at the microRNAs that were downregulated by A2B1 in our A2B1 MCF7 cells and microRNAs that were predicted to regulate PSAT1 and PHGDH by interaction with 3' UTR of these transcripts. And she used different databases to examine this potential interaction. And this is because there were no validated microRNAs targeting PSAT1 or PHGDH. So she came up with 2 microRNAs, miR145-5P and miR424-5P, that putatively target PSAT1, and 2 microRNAs, miR34B-5P and 876-5P, that putatively target PHGDH. And here are the data by real-time quantitative PCR showing that these microRNAs are indeed downregulated in our endocrine-resistant cells compared to MCF7 cells. To validate that PSAT1 and PHGDH are really bona fide direct targets of these A2B1 downregulated microRNAs, what we did was to purchase a dual luciferase reporter vector with either the PSAT1 full-length 3' UTR cloned downstream of the firefly luciferase, and here's the ranilla luciferase, so it's dual luciferase, and or the PHGDH full-length 3' UTR, again, downstream of firefly luciferase. Or we also used PMIR glow vector, into which Kelly Peel cloned the microRNA response elements, and the flanking sequence from PSAT1 or PHGDH into the reporter, again, downstream of the firefly luciferase with the ranilla luciferase, so another kind of dual luciferase reporter. And Belinda did transient transfection experiments in HEC293. So what Belinda did here was to line up the microRNA sequence with the 3' UTR of PSAT1 and or PHGDH. So you can see the recognition between the base pairing recognition of the microRNA and the 3' UTR of its targets. So what should happen if the microRNA really does bind to these microRNA response elements that are present in the 3' UTR of the respective gene, then that will decrease luciferase protein expression. We'll see a decrease in luciferase reporter activity. Namely, we'll see a decrease in the firefly relative to the ranilla luciferase activity in our assays. And so Belinda did lots of experiments, and she found, thankfully, what we were looking for. She was able to confirm that MUIR 145-5P and 424-5P indeed directly target the PSAT1 3' UTR, and MUIR 876-5P and 34B target the PHGDH full-length 3' UTR. And this was also confirmed with using negative controls in that PMIR glow assay as well. And then she also transfected all 4 microRNAs together into LCC9 cells and performed an MTT assay, as we had seen before, where DMSO is our vehicle control, and then 2 different concentrations of 4-hydroxytamoxifen and 100 nanomolar fulvestrant to determine whether the transfection of these microRNAs that target PHGDH and PSAT1 would increase endocrine therapy sensitivity, and the answer is yes. So what we've shown is that hnRNA2B1 decreases specific microRNAs that target PSAT1 and PHGDH, that decreases endocrine therapy sensitivity, hence increasing endocrine therapy resistance. And we published this last year in Endocrine-Related Cancer. Now, I want to say as a little bridge that I started out talking about epitranscriptomics and M6A, and then I got into the A2B1, which is a reader of the M6A. But the serine synthesis pathway is actually connected with methylation of targets because it's involved in S-adenosylmethionine production, because the serine synthesis pathway is linked to the folate cycle in which S-adenosylmethionine is generated. So now this is all new. Everything that I talked about for the first 20 minutes or so has been published, but now we're getting into terra incognita. So I've shown that we had an increase in A2B1, a decrease in these microRNAs that target PSAT1 and PHGDH, and that this increase in PSAT1 and PHGDH is involved in endocrine therapy resistance. So if M6A in the primary or precursor microRNAs is involved in the decrease in these microRNAs with increased A2B1, I asked, will the metal 3 inhibitor, STM2457, so it's a chemical compound, or Si-metal-3 inhibiting, knocking down metal-3 protein expression result in increased miR-145 5P, 424 5P, 34 5P, and 876, and therefore decrease PSAT1 and PHGDH. So, Kelly Peel transfected LCC9 endocrine therapy resistant cells with Si-metal-3 or Si control. And what we can see is that at 48 and 72 hours after transfection with Si-metal-3, we see a decrease in metal-3 transcript expression. And lo and behold, we also see a decrease in PSAT1 and PHGDH transcript expression. So with lower M6A, we see a decreased transcript abundance of PSAT1 and PHGDH. Now, we don't know if this is direct or indirect. There could be M6As in the PSAT1 and PHGDH transcript or not, or something else, an intermediary could regulate PSAT1 that's regulated by M6A, could regulate PSAT1 and PHGDH. So, of course, once you look at the transcript, you also want to look at the protein. So we asked, what happens to PSAT1 and PHGDH protein? First of all, we can see in the LCC9 cells that Si-metal-3 decreases metal-3 protein expression. And we can look at PSAT1 and PHGDH, and you can see that Si-metal-3 is decreasing both PSAT1 and PHGDH protein, both at 72 and 96 hours after transcription. But again, we don't know if this is direct or indirect, because it's likely that the Si-metal-3 affects numerous targets that can regulate PSAT1 and PHGDH transcript and protein abundance. So, for example, some other regulators of PHGDH and PSAT1 transcription are MYC, ATF4, and ATF3. But we had the story of microRNAs regulating PSAT1 and PHGDH expression. So going back to the hypothesis, Si-metal-3 will increase miR's 145 5P, 424 5P, 34 5P, and 876 5P, and decrease PSAT1. So what we can imagine is that with M6A, we get a degradation of the microRNAs, and therefore we relieve inhibition of PSAT1 and PHGDH, and so they would increase. But if we remove M6A by inhibiting or knocking down metal-3 expression, we would expect an increase in these microRNAs and therefore a decrease in PSAT1 and PHGDH. So no more M6A, miRs are more stable, less PSAT1 and PHGDH protein. So what did we see? So these are the cells, the LCC9 cells that we just saw, that we saw that Si-metal-3 was knocked down both at the transcript and the protein levels. And what did we see with 145 5P? Well, in fact, we saw an increase in miR-145 5P with Si-metal-3. So that would go over here. We get an increase. We would expect less PSAT1 because miR-145 5P targets PSAT1. Now, miR-424 also targets PSAT1, but it was not increased. In fact, it was decreased with Si-metal-3. So here we'd expect more PSAT1. So this disagrees with my hypothesis. And on the PHGDH side, what do we see? With knockdown or Si-metal-3, we see an increase in miR-876 5P and an increase in 34B. So we would expect less PHGDH with more microRNA protein should go down. And what do we see at the protein level? Well, we know that we saw a decrease in PSAT1. So while the miR-424 result disagrees with the hypothesis, the 145 5P agrees with the hypothesis, and the data with the miR-876 5P and 34B being increased with Si-metal-3 agree with the decrease in PHGDH protein and transcript expression that we saw in the LCC9 cells. So the next question I asked was, does the metal-3 inhibitor STM2457, and here it's its chemical structure, and we're using, I'm not showing the data, but Kelly has done a number of experiments to look at the IC50 for all our breast cancer cell lines. And it turned out that we're going to use 1.6 micromolar, which is 25% of the IC50 value that we see in LCC9 cells. So we don't want to kill the cells, but we want to inhibit metal-3 activity. And these experiments were performed by Kelly with Raya Hewlett, who is a junior biology major pre-med student and a swimmer on the UofL swim team, who got a gold at an NCAA event fairly recently. So what do we see? So this is another MTT assay, and I've plotted it a little bit different. I'm showing these little bars instead of just the dots because I thought, well, maybe this is a better way to present these data. So these are LCC9 cells, so we know they're not growth inhibited by 4-hydroxytamoxifen or fulvestrant. What happens with STM? Well, that should be 1.6. That's my bad typo. But it's actually stimulated compared to the DMSO vehicle control. So I don't know why that would be, but interesting. But if we combine the 4-hydroxytamoxifen at the two different doses with STM2457, we see an inhibition of cell viability relative to control. And the same thing is true with fulvestrant. So we see a sensitization of the LCC9 cells to both 4-hydroxytamoxifen and fulvestrant in the presence of STM2457 treatment. Right. So, okay, that's it. All right. So then we saw that SI-metal-3 inhibited PSAT1 and PHGDH expression. Well, with the metal-3 inhibitor, STM2457 result in decreased PSAT1 and PHGDH. And the answer is absolutely not. In fact, we saw the opposite. So we treated the cells again with the same concentration, 1.6 micromolar STM2457, and we're getting a time-dependent increase in PSAT1 and PHGDH transcript expression. So this contradicts our hypothesis, and it's the opposite of SI-metal-3. Well, what about protein? Maybe something's different here. Oh, and this I was just working on this afternoon, because we also have, as you will soon see, differential gene expression by Illumina short-read RNA-seq of these MCF7 versus LCC9 cells. And so I looked at what happened, and we treated some cells with STM2457 for the RNA-seq analysis. And what we can see here is that we see an increase in PHGDH and PSAT1 in the MCF7 cells treated with STM2457. So these data are in agreement with what we see in the RNA-seq data. Well, what about protein? Same thing. We see an increase in protein with the STM2457. So it's opposite of that SI-metal-3. So I really don't know what's going on. So I'm sharing with you data that I can't explain yet. Is it off-target effects? And which are the off-target effects? Is it STM2457 that has off-target effects that are increasing PHGDH and PSAT1? Or is it the SI-metal-3 that is having some off-target effects such that I'm seeing a decrease in PSAT1 and PHGDH? But I will say that these data that with the STM2457 agree with a publication in leukemia cells in which the investigators deleted ALK-BH5. That's one of those M6A erasers. So if you decrease the M6A eraser, you get an increase in M6A, and they saw a decrease in M6A stability of PHGDH and PSAT1. So these data would agree with those, but those were leukemia cells. So maybe it's something about stability, right? M6A is involved in stability of RNA. So how stable, I asked, are PHGDH and PSAT1 transcripts in LCC9 cells? And so we treated the cells with actinomycin D, which is a transcription inhibitor, and at that concentration, 5 micrograms per ml, at time 0. And we also treated the cells either with DMSO or 4-hydroxytamoxifen in case there was any effect of 4-hydroxytamoxifen on stability in the cells. And we found, lo and behold, wow, PSAT1 and PHGDH are very stable in LCC9 endocrine-resistant cells. What about protein? Well, to look at protein stability, you treat the cells with cyclohexamide, which inhibits translation. And so what we can see here is that there's no loss of protein. And if anything, we're getting more stabilization with cyclohexamide. So these are very stable transcripts and proteins in LCC9 cells. So coming back around to the microRNAs that we have proven directly target PHGDH and PSAT1, I asked the question, what about STM? Does it affect the microRNAs? So before, what we saw was that with metal-3 knockdown, we saw an increase in microRNA and a decrease in PSAT1. Now, STM2457 is inhibiting metal-3. What'll happen? Well, it's the opposite of what you think. We showed that PSAT1 was upregulated by STM2457, and in fact, the microRNAs, miR-145-5P and 424-5P, are inhibited over time by STM2457. So these results fit what we see with STM2457 having the opposite effect on PSAT1 and PHGDH compared to SI metal-3. So we have a mystery and a concern. Why is our SI metal-3 and STM2457 having opposite effects on microRNA and their target PSAT1 and PHGDH? So maybe it's something in between these events that I need to look at. So soon we're going to be examining MYC, because MYC regulates these microRNAs as well as PSAT1 and PHGDH and ATF4. So maybe I'll learn something from that. But as of yet, I really don't know. So summary 2, we showed that SI metal-3 reduced metal-3, PSAT1, and PHGDH protein transcripts and protein abundance. SI metal-3 increased miR-145 that targets PSAT1 and miR-34B and 5P and 876-5P that target PHGDH. The metal-3 inhibitor STM2457 increased sensitivity of LCC9 endocrine therapy resistant cells to 4-hydroxytamoxifen and fulvestrant, so it restored some degree of endocrine therapy sensitivity. But that same metal-3 inhibitor, STM2457, increased PSAT1 and PHGDH transcript and protein abundance in the LCC9 cells and decreased miR-145, 5P, and 424. We did not, and were unable to detect miR-876, 5P, and 34B, 5P that target PHGDH in the STM2457 treated cells. So we'll have to do something a little bit different to get at what they do to the PHGDH targeting microRNAs. And we found that PSAT1 and PHGDH transcripts and proteins are very stable in LCC9 cells. And again, I don't understand the reason for the opposite effects of SI metal-3 and STM2457 unless it's off-target effects. So what am I going to do next? I'd like to co-treat LCC9 cells with STM2457 or transfect with SI metal-3 and treat with actinomycin D or cyclohexamide and look at transcript stability and protein stability in the cells. And I want to transfect antisense miRs in SI metal-3 transfected cells to see if this blocks the decrease in PSAT1 and PHGDH transcripts and proteins, because that would identify those microRNAs as M6A regulated by metal-3. So now another new topic. And this is looking at the position of M6A in mRNA transcripts as potential drivers of endocrine resistance in breast cancer cells. So Kelly Peel treated MCF7 and endocrine therapy sensitive and LCC9 endocrine therapy resistant cells. First, the cells were serum SAR for 72 hours, and you have to do that with breast cancer cells in order to see any effect of hormones or endocrine therapies on the cells. And then the cells were treated for 24 hours with a vehicle control, DMSO, 100 nanomolar 4-hydroxytamoxifen, which is a concentration of 4-hydroxytamoxifen that is found in patients' tumors who are taking oral tamoxifen citrate. Oh, and that's a micromolar, 1.5 micromolar STM2457. And we prepared and ran 5 biological replicates per group. And this is Kelly actually loading the nanopore MinION device with the RNA. And how this works is, so this is direct RNA sequencing. So we have this nanopore with a motor that pulls the RNA through, and an electrical signal is generated that can be read as the sequence. And also a different electrical signal is indicative of an N6 methyl group by processing the data. So I'm just going to say this has been a long experiment. So we started isolating RNA for this experiment in May 2022 and sent samples starting in August to Dr. Rushka of August of 2022. And ultimately, we got the M6A net report from Dr. Rushka in January of 2024. The time required for these experiments and the cost is really something. So I'm very, very grateful for all the financial support from grants that have allowed us to do this work. Now, when Dr. Rushka first started to analyze the data, he used M6A net, which uses signal intensity and sequence features. And this is the publication from which he got the program to examine the data. But he and Belinda Petrie have spent an enormous amount of time figuring out the best way and correcting the scripts in R to make this work. And the inputs are the FASTA5 files for signal intensity and the FASTQ files for the reads. And the output is probability of an N6A modification and the modification ratio. So first question is, what is the M6A distribution in gene and transcript? So this is what Belinda has put together, and here she is at the hooding ceremony in December. And what we can see is we have MCF7 treated with DMSO or 4-hydroxytamoxifen, LCC9 treated with DMSO vehicle control or 4-hydroxytamoxifen. We have the number of genes with M6A modifications, the average M6A per gene, number of transcripts with M6A modifications, and the average M6A per transcript that you can see here. And the M6A net analysis confirmed that the M6As were located in the Dratch motif. So remember I said there was that A that's going to be the target of metal 3, putting that methyl group on, and it's always invariably followed by a C. So you can see there's the Dratch motif in all the samples. And then Belinda put together a Venn diagram to look at those transcripts that are common versus different in the different treatment groups. And so we can imagine that these 85 M6A decorated transcripts might be involved in endocrine sensitivity, and these might be involved in endocrine therapy resistance. And we'll get into that as we go on. So then the question becomes, are there differential M6A modifications between the endocrine sensitive versus endocrine resistant cells? And Belinda has various ways of analyzing the data, and I'm just showing this one as one way in which the data are compared. So now we're comparing groups, so we can compare MCF7 cells treated with 4-hydroxytamoxifen versus the vehicle control. And we see 85 different M6A changes, 71 cases where there's an increase in M6A in a target transcript, and 14 in which there's a decrease. And you can just look at this whole thing. And we see the most transcript changes in MCF7 4-hydroxytamoxifen treated versus LCC9 4-hydroxytamoxifen treated. And we also see changes between the LCC9 4-hydroxytamoxifen treated and versus the vehicle control, and STM2457 treated cells compared to vehicle control or to 4-hydroxytamoxifen. And interestingly enough, with the STM2457 treatment, we see both increased M6A and decreased M6A, so that there could be an intermediary target for STM2457 inhibition of methyltransferase activity. That's the transferase activity, because we're using a 24-hour treatment. So I wanted to know if STM2457 decreased known M6A regulated transcripts. So a big one that people have reported since about 2016 is MYC. It has a number of M6A modifications in the transcript. And in fact, we can see that we saw a decrease in MYC with STM2457 treatment of MCF7 cells. So yay, that means that STM2457 is doing what we would expect based on the literature. And then I found some other gene targets that are regulated by M6A methylation in the literature. We have 3 in agreement where we see a decrease with STM treatment of MCF7 cells, and 3 cases where there's not agreement. In fact, we see an increase here in TAZ, in MIB, and SOX7-2. But what I didn't put in is that these are in different cells. Nobody has looked at this in MCF7 or other breast cancer cells. These are in leukemia cells or melanoma cells or prostate cancer cells. So then Belinda made a volcano plot of the differential M6A modifications in MCF7 cells treated with the metal 3 inhibitor STM2457 versus MCF7 treated with the vehicle control. And I know it's probably hard to read, but there's a lot of EEF1A, and that's a protein that is involved in translation. So there are over 16 modification changes up here. So she filtered out EEF1, and that allowed us to expand this bottom range. And interestingly, previous studies have used EEF1A as a positive control for M6A sites, just as I mentioned MYC and EGFR and the other transcripts in the previous slide. So that means that metal 3 inhibitor STM2457 reduces M6A in translational components, which makes sense because M6A is a modification that regulates translation. And then Belinda performed metacore analysis of the M6A modifications in the MCF7 cells treated with the STM2457 versus control to look at pathway maps. And you can see things like transcription, negative regulation of HIF1 alpha function, and process networks. And look what came up. Signal transduction, ESR1, estrogen receptor alpha, nuclear pathway. And the numbers, which are very tiny at the moment, are the number of transcripts that are differentially M6A modified in the STM2457 treated versus vehicle control of MCF7 cells. And then she did metacore analysis of differential M6A modification between MCF7 versus LCC9 cells, and looked at protein pathways in metacore and process networks. And you can see some differences there, for example, protein folding. And I'm just going to kind of bust through this. And in parallel with the direct RNA sequencing by nanopore, we did short, so that's long read sequencing. I neglected to say that, but we also did short read RNA sequencing with Illumina on the very same samples, again, with the same number of replicates, 5 replicates per cell line and treatment group, to identify differentially expressed genes. And so here are the numbers of differentially expressed genes comparing different samples, and you can see the most differences between LCC9 versus MCF7 cells, whether treated with vehicle control or treated with 4-hydroxytamoxifen, and that's what's indicated in these volcano plots as well. We also asked the question, are there differences in the M6A writers, readers, and erasers? And what we can see is that in LCC9 cells, there is a decrease in YTH-DF1, YTH-DC1, and an increase in, excuse me, IGF-BP2. And likewise, there's a decrease in 4-hydroxytamoxifen-treated MCF7 cells of IGF-BP2. And I have written, or maybe Belinda and I have written some things about these writer proteins down here. Again, the role of the writer protein is very cell context dependent, and these readers also interact with other proteins. So whether they increase translation or stability is very cell type and context specific. Now, just today, and literally, honestly, about 10 minutes before I came down here, Kelly Peel sent me some data on Westerns that she ran overnight in that a new undergrad in the lab. Tyler Brown and I were rinsing and putting into secondary antibody this morning. Okay, and what she has done, Kelly has done, is to confirm that YTH-DF1 is downregulated at the protein level in LCC9 cells, and IGF-BP2 is upregulated in LCC9 cells. So that agrees with the transcript abundance that was detected in the differential gene expression that Belinda figured out. And so here's a heat map then comparing MCF7 versus LC9 cells treated with vehicle control and lots of differential genes and pathways. And then if we can compare 4-hydroxytamoxifen treated versus vehicle control treated, up came one nuclear receptor, NUR77. And then we can compare LCC9 cells treated with 4-hydroxytamoxifen versus control. One interesting gene I can see right off the top, FGR, which is involved in endocrine therapy resistance. Oops. And then here are the heat maps for MCF7 cells treated with vehicle control versus the metal 3 inhibitor, STM2457. And what you can see here, I've circled the particular gene amigo-2 that we're going to look at more closely. So amigo-2 is interesting because it has decreased M6A with STM2457 treatment compared to vehicle control. So that means it seems to be a target of metal 3 transferase activity. And it's also decreased in the 4-hydroxytamoxifen treated cells. The Metacore analysis of the differentially expressed genes in MCF7 cells treated with STM2457 brought up Wnt signaling and beta-catenin signaling pathways, and these are known to be altered in endocrine-resistant cells. And then, so we've been looking at M6A in genes and differentially expressed genes. What Belinda has done with considerable effort is to integrate the differential gene expression with the differential M6A modification. So first, the comparison is LCC9 versus MCF7 cells just treated with vehicle control. And so these are the genes that are different between LCC9 and MCF7 cells and have differences in M6A abundance in transcripts. So 29 have an increase in M6A and an increase in gene transcript abundance. 10 have a decrease in M6A, decrease in gene transcription. So you can look at all that. And then, so interestingly enough, when we look at the Metacore analysis process network, we see a number of cytoskeletal and cell adhesion process networks. And endocrine-resistant breast cancer cells exhibit altered cell matrix adhesion and highly migratory phenotype, which may impact tumor spread in vivo. There's amigo-2 now looking at the top 20 genes in LCC9 cells with the highest number of M6A versus MCF7 cells. And the top 20 genes in DMSO, in LCC9 cells treated with DMSO that have decreased M6A compared to MCF7 cells. So Amigo-2 is at the top of our list. It has 26 M6A sites in 3 different transcripts, so an average of 9 M6A modifications per transcript. And I know we're getting close to time, so Belinda has also put together the distribution of differentially expressed genes with changes in M6A modification, comparing the 2 cells where we have M6A up and the gene up. So here's a list of genes. You can look for your favorites. M6A down, gene down. And then top 20 transcripts downregulated in LCC9 for hydroxytamoxifen versus MCF7 cells. And there's Amigo-2. And also in this list was NR2F2, and we published a lot. And NR2F2 is the orphan nuclear receptor CoopTF2. And we published a lot about CoopTF2 and breast cancer. So it's nice to see CoopTF2 on the list as well. So Amigo-2 is M6A methylated in MCF7 cells treated with 4-hydroxytamoxifen, but not in the LCC9 cells. And the transcript abundance is lower in the LCC9 compared to the MCF7 cells. So what's Amigo-2? It has a cool name. It stands for adhesion molecule with Ig-like domain. And it's located either in the plasma membrane or in the cell nucleus. And it's involved in programmed cell death and apoptosis. It was first identified as a novel gene that has a role in metastasis, and high Amigo-2 was a prognostic biomarker of prostate cancer correlated with docetaxel resistance. If we look in KM plotter, we can see that high Amigo-2 transcript is significantly associated with reduced relapse-free survival in all breast cancer, so it seems to be potentially oncogenic. However, Amigo-2 was downregulated and was one of the 4 drug resistance related genes in patients with ER-positive, HER2-negative breast cancer, and that would mimic our LCC9 cells. So what's Amigo-2 doing? So Bailey Valdez is currently an M2 student. She was supported by the NIDDK T35 grant and worked in my lab last summer, and she performed this Western blot. And we can see that the protein levels of Amigo-2 are also lower in LCC9 cells compared to MCF7 cells. So Belinda analyzed the location of the M6A within Dratch motifs in the 3, remember I said there were 3 transcripts of Amigo-2. So these are the 3 transcripts, and you can see where the M6A sites are. They're all at the end of the transcript, I think. So what's next? Well, we need to confirm that Amigo-2 mRNA is a direct target of metal-3. And so what I want to do is take Amigo-2 mRNAs, the coding sequence plus the 3' UTR, with either wild type or mutant M6A within that Dratch motif. And I can insert that downstream of luciferase reporters. I showed that dual luciferase reporter, so we can put it downstream of the Firefly luciferase. Transfect HEC293T or MCF7 cells with this reporter with the Amigo-2 track containing sequences in it, and look at reporter gene, and then I can transfect the cells with a metal-3 expression vector and look at stability of the transcript itself, the metal-3 transcript itself, and look at luciferase activity as an assay. So summary 3, we identified differentially expressed genes and M6A differences in endocrine sensitive versus resistant MCF7 and LCC9 cells. We confirmed that STM2457 reduced some known M6A regulated transcripts in MCF7 cells. We identified Amigo-2 as an M6A regulated transcript with higher expression in MCF7 than LCC9 cells. So what I think is going on is that maybe M6A is increasing amigo-2 transcript stability and translation. And since it's involved in apoptosis and endocrine therapy sensitivity, that would be a good thing, because maybe you need amigo-2 to send the cells to apoptosis in response to tamoxifen treatment. Whereas there's less M6A in amigo transcripts in LCC9 cells, so the protein is not translated, or the transcript is less stable, so we don't have amigo-2, so that may lead to apoptosis resistance. So we have experiments yet to do to examine this hypothesis. So I'm very grateful to Belinda Petrie and Kelly Peel, to Ollie Wilt, who's an M4 student who's doing distinction in research, and she was in the lab doing some stability experiments with Kelly this past fall, Bailey last summer. Laura Winkler, Rai Utal are currently undergraduate students in the lab, and Maddie Whitworth is a high school student at Assumption High School. And of course, Eric Grushka, who's now a postdoc mentor for Belinda, is greatly involved in all the bioinformatics on this project. That's it. And today's code. There it is. Oh, so I have to get out of here and slideshow. Slideshow. Stop sharing. Well, let's see if I go. Oh, what if we do this? Slideshow. Oh, there it is. Okay, now blue thing. There we are. Chat. Do you have any knowledge about testosterone? Nope, I don't, but I could look it up. Oh, there's a question. Sorry, I thought it might show up there. Do you have any knowledge about the microRNA mediated mechanism of lowering testosterone resistant prostate cancer? So there must be specific microRNAs that are involved in androgen resistant or castration resistant prostate cancer. I don't know what they are, but I bet they're known. You could look them up in PubMed.
But the M6A, it's been on many other cells, right? It's present and is diffused there. So what is its actual role? And like, if you're trying to manipulate it in these particular cells, but if I use something as a pill, and like trying to,
Right.
what's going to happen?
Right. Yeah. So that's a problem, right? We know that STM2457 in a dose-dependent manner inhibits the viability of breast cancer cells. We are still working on whether the endocrine therapy resistant cell lines are more or less sensitive to STM2457. Those two experiments, we need to do a third to get the variability down to make a firm conclusion. But the hope would be we could do something to target, specific microRNAs. Now, of course, there's DCAS13 and guide RNAs that we can use to modify specific RNAs. So maybe something along those lines.
So is there anything that we know that regulates these things?
Yeah, I don't know what epidermal growth factor signaling and so the question is, well, what regulates like metal 3 or some of the erasers, ALK, BH5, FTO? Obviously, they're regulated at the transcription level, but they are also regulated by MAP kinase signaling, for one thing. So by phosphorylation, by acetylation as well. So. Yeah. All I can do is try to look at stability, and what's going on, and maybe do time course experiments with STM2457. Boy, then we'd have to do some kind of RNA-seq to find out what it's targeting over time. What's it targeting 1 hour and 6 hours, or back off to some shorter period of time and look then at whether PSAT1 and PHGDH are regulated. Yeah, and the microRNAs as well. Thanks for coming.
Hi, Carolyn.
Hi.
You
Sorry,
you
there's another
mentioned
question.
that you have some unpublished data on in your grand rounds. So
Yeah.
we would like to, Dr. Mokshagandan and I are going to co-edit a special issue in the Frontiers in Endocrinology. And I think we would like to very much invite you to submit a manuscript in that regard.
That'd be great.
We will send you the invitation shortly.
Thank you. Glad to help with that. Thank you. Can HUVEC cells also be used as ET-resistant, ET-sensitive cells for this kind of study? I don't know. So I know that we've done work with HUVEC cells, but that we were really looking at membrane estrogen receptor, which is enriched in the HUVEC endothelial cells. So I don't know whether you could use them as in those studies. Thanks, folks.